demonstrate how to use circosVCF in order to generate a circus plot from a VCF file containing data from multiple samples. Please note that circosVCF can also process one or several VCF files for single samples. Here we will use data that was published by the Robinson Group. The paper provides exome data for 4 family members, parents, and their daughter and son. The VCF file itself can be downloaded from this link. Each individual in the file has a unique ID. For example, the ID that ends with 2015 represents the son, and so on. The aim of this analysis is to find out who is most similar to the father, the daughter or son, and also to visualize the variance establishing this similarity. So let's see how this can be accomplished with circosVCF. Let's start. The first step is to add a genome size information. It can be uploaded from a local file or added manually by entering chromosome names and their length. But the easiest way, especially for model organisms, is to load the data directly from UCSC. First, select the organism, in our case, human. Second, select the relevant build, HG19 for our VCF, and click Save. By default, all chromosomes are selected and therefore will be plotted. We will first deselect all chromosomes and select only chromosome 1 for plotting, and then click Save. If you wish to add annotations such as gene symbols to the plot, select the Annotations option. Again, let's select Human and HG19. Now it's time to upload the VCF file. Define the rings that will appear in the plot. The first ring will represent the daughter genotypes that are identical to her father. So let's call this ring Daughter. 2 options are available for representing SNP information. The first is the calculation of number of SNPs within a defined genomic interval, where darker color on the plot represents denser regions. The other option is to draw a line in each SNP location, where each line is colored based on its genotype. In our demo, we will use the first option. Additional information can be retrieved on each analysis step by clicking the question mark. Click Next. Since our goal is to view the similarity between daughter and her father, we would like to find SNPs that abide all 3 conditions, which can be defined in a condition tree using AND logic. The first condition is that the daughter genotypes should equal her father's. Hence, we require equal columns and select those representing the daughter and her father. The second condition selects daughter genotypes that are not equal to her mother's. Similarly, we will select for daughter genotypes that are not equal to those of the son. Click Save. We can edit ring parameters by right-clicking the ring name and selecting Edit. For example, let's increase the ring diameter to be 20% of the entire plot diameter. A second ring will represent the son's data. We copy the daughter ring and edit it. First, we will change the name and update the conditions so that those will replace the daughter ID with that of the son. Now we can send the job to be processed on a remote server. We will enter our email information and click Start. A link to the final plot as well as to a file describing the number of SNPs found on each ring will be sent to our email and available for downloading for 3 days. Once we get the email, we can check the number of SNPs unique to the daughter compared to the son. Also, we can view the circus plot itself.